Deep within the gut's epithelial layer are specialized sensory cells that convert mechanical stimuli into electrical signals. They convey this information to nerve cells in the gut and those that relay information to the brain, acting like touch sensors in the skin to transmit gut feelings. Researchers study the neuroscience of gut touch to learn more about what these signals mean and their role in gastrointestinal health and disease. Welcome to The Scientist Speaks, a podcast produced by the Scientists Creative Services team. Our podcast is by scientists and for scientists. Once a month, we bring you the stories behind newsworthy molecular biology research. In this episode, Iris Kulbatski from The Scientist spoke with Arthur Beyder, a gastroenterologist and biomedical engineer at the Mayo Clinic, to learn more about his team's research on the emerging neuroscience of gut touch.
The gastrointestinal tract is a hub of activity and, in many cases, a rather touchy subject. Arthur Beyder is fascinated by the gut's mechanosensory system, the specialized sensory cells, similar to skin touch sensors, that detect the physical textures of food particles. Known as gut touch, this system organizes the distribution of food particles in the small bowel and manages stool size in the colon to ensure optimal digestion. Beyder and his team traveled a long and winding road to understanding how these cells work and their importance in gut health and disease. His research began with a simple yet fascinating scientific question. Does the gut have a built-in mechanism to feel and assess the physical qualities of ingested food, much like the fingertips? The answer, he found, was yes.
We have a sense of touch in the skin that we use to really interface mechanically with the world around us. Before we had forks, knives, and spoons, we would use our hands. And touch in the fingertips was extremely important to get us a sense of the quality of food. If it's slimy, maybe it's rotten. If it's squishy, maybe it's not fresh. If it's too hard, we'd be careful in terms of mastication in the mouth. This is a critical area in terms of central neuroscience and somatosensory activity. We eat it as physical food along with liquids. All of that physical stuffs, all of that bulk is then taken by the stomach and the small bowel and processed mechanically and chemically. The lining of the gut wall, it's kind of like the skin of the gut. There's specialized sensory cells that express the same mechanical sensors as the Merkel cells, which conduct touch in the skin. Merkel cells are these little epithelial cells that hook up to the neurons. They're responsible for feeling the mechanical forces when you touch something to the skin. And you have an equivalent of that kind of a cell called enteroendocrine cell that expresses the same kind of mechanosensor in the gut as it does in the skin. Since then, we've found receptors on the gut lining that are like these Merkel cells. We're finding some neurons that are mechanosensitive built into the wall of the gut. And this is something that endows the gastrointestinal tract with unique functionalities.
Beyder is fascinated by the basic anatomy of the gut and the ways in which it does its job, describing it as separate from the rest of the body to a certain extent. From a comparative anatomy perspective, Beyder describes the gastrointestinal system as highly conserved, having evolved in lower organisms before higher order sensory processing did. He also draws physiological comparisons that consider touch sensing overall and how pathophysiological changes in specific touch systems of the body can inform others. In this way, gastrointestinal abnormalities may provide important clues to understanding other disorders.
Our gut is kind of like our body's inside out, with the gut wall being on the outside. And the gut epithelium, the skin of the gut, is like the skin that we have. It might not go to conscious processing, but we conduct all of this digestion and all this particulate sensing technically outside the body, separated by the gut epithelium. That really twisted me inside out. No pun intended on this. I really like the idea of drawing pathophysiology comparisons. Autism is a really interesting example. Autistic kids are well known to have tactile hypersensitivity. They really struggle with touch. huge number of autistic kids have GI abnormalities, constipation, or other things. I'm wondering, hey, maybe along with the touch system that's disrupted in their skin, maybe they have disruptions in gut touch. And maybe that's a part of the reason why they have GI problems, which have thus far eluded us. I love this kind of thinking because it allows us to start drawing parallels and really start thinking outside the box about some of the very common conditions.
Much like the other ways in which the gut acts as a second brain, gut touch happens without the need for central nervous system processing. The gut's ability to detect and handle food texture depends on the enteric nervous system and is automatic, beyond conscious awareness. Gut touch, as a tactile sense, plays an important role in healthy gastrointestinal function by redistributing food particles in the small bowel for better digestion. Gut touch in the colon also helps identify stool size and manage waste disposal. When these processes are disrupted, poor motility can lead to constipation and other problems. Beyder and his team also implicate disrupted gut touch in functional gastrointestinal disorders, such as irritable bowel syndrome.
We are trying to figure out exactly what that system is there for. Thus far, we've settled on the following. Once you eat a meal, the stomach will grind it up with gastric acid and pancreatic juices, but foods are not fully ground up. And while they're not ground up, you're not able to extract all the nutrients. The gastrointestinal tract's main function is to suck out every single nutrient, every single water molecule, because we did not always live in the world of plenty. If it gets particulates, which are physically not digested enough, it might slow things down and kind of keep grinding them up. That's exactly what we see with the gut touch system. When I was in med school, we learned that majority of digestion and absorption happens in about the first 10% of the small bowel. But small bowel is 6 feet. What's the other 5.5 feet or so of the small bowel for? And our hypothesis is that it's there to, in at least part, continue to work on these particulates to grind them down and extract all the nutrients, engaging the entire length of the small bowel rather than just small regions of it. Gut touch in the colon is important for detecting the size of the stool. If we disrupt this system, the colon and the small bowel forget how to deal with particulates. They're not as effective in terms of redistributed things, packaging things. This system, if it's broken, it'll likely affect digestive motility from the small bowel and excretion motility in the colon.
The cells responsible for gut touch make up a relatively small percentage of the gastrointestinal epithelial layer. Embedded in the membrane of these cells are specialized ion channels, named piezo 2 channels, that sense and respond to mechanical stimuli. Physical forces exerted onto the gut wall's epithelial layer activate these channels, allowing them to transmit information about such forces to adjacent nerve cells through changes in calcium ion gradients. These channels are also present in other organs that rely on mechanosensation, and their discovery by neuroscientist and molecular biologist Artem Pataputian and his team at Scripps Research earned him a Nobel Prize. Beyder and his team are particularly interested in the molecular signals that occur after piezo 2 channels are activated in the gut, which happens within milliseconds.
We're interested in processes that amplify calcium and culminates in release by exocytosis of signaling molecules. The big classical one that we are following in the gut is serotonin. Upon stimulation of the piezo channels, serotonin will signal to essentially anything that's got serotonin receptors. We're following up on neurons that do this. Intrinsic gut neurons built into the wall of the gut. Their cell bodies are right inside of the gut wall. They have serotonin receptors. They respond to gut touch sensors and they modulate motility. If you were to take the gut out, scratch the mucosa, it will perform all of its functions as if it's still inside the body. There's this built-in system that has an epithelial and a neuronal connection that likely is responsible for gut touch. Gut touch cells also connect to neurons that lead outside the gut. We're finding them, as I described, in the small bowel, sort of regional, but also distal parts of the colon converse with more proximal parts of the gastrointestinal tract, like the stomach and the small bowel. That's the traffic control that allows you to take 12 feet worth of plumbing and regulate it at all the different checkpoints. Most of us don't appreciate the gut touch system. It's supposed to, I think, work underneath our conscious understanding.
Beyder and his team were interested in knowing the exact position of gut touch mechanical sensors in epithelial cells and how exactly they transduce force. They hypothesized that the sensors were located at the top of cells, acting like a doorway in response to mechanical pressure, but were surprised to find them on the side of cells instead. This made the scientists wonder what the cells were doing there and how exactly this position helped them perceive force. Knowing where the mechanosensors were situated, Beyder and his team began plucking them out of gut touch cells to determine whether they were tethered to any other proteins that may play an important role in sensing physical forces. They relied on high-resolution imaging to localize structures, calcium imaging to assess function, and co-immunoprecipitation to determine associations between piezo2 channels and other proteins. They found that along with the piezo2 channels, they were able to pull out actin and E-cadherin.
Actin is well known as a cytoskeletal element, important for structuring cells, very important for epithelial cell structures. E-cadherin is also well known, part of the junctional complexes between these epithelial cells. If we disrupt E-cadherin, actin, we have a very different mechanical response of the gut touch sensors that is significantly blunted. The kinetics are all wrong. We are now asking whether or not these channels are wired in mechanically. Are they little tethers that pull on them? Or if they're there because of positioning in the right mechanical space so that they can just feel the force. These molecules don't live in isolation. They are parts of major complexes, and those complexes are built in such a way that disruption of any of the pieces leads to problems. The most fascinating bit for me is that you can have a mechanosensor expressed in a cell, but if you happen to monkey around with one of its connecting pieces, you can disrupt significantly mechanosensory cell function. We believe that the sides of the cell of this gut touch mechanosensor are like springs in a mattress. So if one were to lay down on a mattress, the majority of force ends up in those springs because of that force distribution. The epithelium is almost like a mattress with springs that run from basal side to the apical side, likely being the carriers of force that's input on that epithelium. So it would make sense then to put mechanosensitive elements into that region. Junctional complexes, tight junctions, adherens junctions have mechanosensors. That is where they put. And now we have an important specialized mechanosensor, gut touch one, that is in the same place.
As a physician scientist, Beyder is especially motivated by the translational aspects of gut touch research and the ability to offer new therapies to patients that address deficits in gut mechanical sensing. For example, other researchers have developed mechanical therapies to target chronic constipation that makes use of a small vibrating pill. Now FDA approved, Beyder hypothesizes that this approach works by activating gut touch and stimulating colon motility.
I had set off on my career as a physician scientist, really hoping to refine and expand the mechanopathology field and mechanotherapies. Lots of patients from my clinic have issues with mechanosensory processing. Our tools right now and our therapies are rudimentary. I would love to understand mechanisms of processing between luminal things that we eat, which set off symptoms, and how molecularly, cellularly, circuit level are these things processed. Once we have that, we can begin to see which of the cell types and molecules are disrupted. So it'll give us a roadmap to target things both diagnostically and therapeutically. And I would love to introduce mechanical therapies. I'd love to expand the understanding of where gut touch is disrupted, whether it's autism, chemotherapy, neuropathy from diabetes, or other things where you have overlap between skin and gut conditions. I'd love to understand the mechanisms, and I'd love to have the diagnostics and therapeutics that would target that.
Thank you for listening to The Scientist Speaks. This episode was produced by the Creative Services team for The Scientist and narrated by Iris Kulbatski. Please join us again in June as we learn how researchers explore the potential of nanotechnology to target cancer. To keep up to date with this podcast, follow The Scientist on social media and subscribe wherever you get your podcasts.